Okay, so our presentation is going to be about the extensions and modifications of basic principles. So first we're going to talk about the classic paper, which was the genetic basis of Haldane's rule. And in order to know what we're kind of talking about, we have to talk about what Haldane's rule is. So it states, when in the F1 offspring of two different animal races, one sex is absent, race, or sterile, the sex is the heterozygous sex. So in this paper, they talk about three possible explanations for Haldane's rule. So the first one was males are innately more sensitive than females to the effects of hybridization. And basically Haldane in that paper just says that's not possible. They already disproved that one. The second one was the explanation that both Haldane and a few other scientists gave, and it was that the heterogamic infertility in hybrids arise from a genetic imbalance between the X chromosomes and the autosomes. And the third explanation was this syndrome is caused by the mismatch of an X and Y chromosome. And that is what the author of this paper, Coyne, is going to test and kind of figure out if that's a possible explanation for Haldane's rule. So he did two different tests throughout this paper. And the first one was to test the X autosome imbalance theory, which is the one that Haldane originally supported. So in order to do this, he produced interspecific hybrid females that would receive both X chromosomes from a single parent species. And the females would then have the same amount of X autosome mismatch as a sterile male. And according to that X chromosome and autosome theory, you would expect there would be sterile females. However, the results, as you can see from this bottom table, is that there was no significant decrease in fertility of these females. So it shows that the second explanation, the one that Haldane thought could be a possibility, is actually not a possibility for the explanation of his rule. The second experiment he did was when looking at fruit flies, he did two different crosses. There was cross A, as you can see in this bottom figure, and it had completely discordant X and Y chromosomes. And it showed that 5 out of the 615 fruit flies they used had motile sperm. And then there was also cross B that had a mixture of X chromosomal matter from 2 species of fruit flies and a Y chromosome from only one of the species. And this had 65 of 682 had motile sperm. So these results were shown to be significant, supporting the third explanation that was shown previously, saying that the infertility of the fruit fly seems to arise from the genetic interaction between the X and Y chromosomes rather than an imbalance as previously thought between the sex chromosomes and the autosomes.
So I am going to be talking about our found paper, which is titled Haldane's Rule in the 21st Century. Haldane's Rule, or HR as they titled it, has been confirmed in many groups such as Diperta, Mammalia, Orthoptera, Telosti, Amphibia, Aves, Leptopterta, and Reptilia. The two main theories of HR are dominance theory and the faster male theory, which I will be talking about a little more on the next slide. Throughout the past 15 years since this paper has been published, researchers have shifted their focus to the lesser known groups of Haldane's rule, which are the amphibia. By studying these lesser known groups, researchers have discovered new insights for testing HR. Of the 85 studies that they conducted, around 20% of them had patterns that were not predicted by Haldane's rule, and these can be explained by postulating cytonuclear incompatibilities. So the first theory that I'm going to be talking about is the dominance theory. This theory states that if there are BDM incompatibilities between the genes, the autosomes, and the sex chromosomes, then the F1 homogametic generation will be heterozygous for all of the genes. They will also go through a fitness loss, but they will only lose fitness for the X-linked genes that are dominant. In 1997, Torelli and Begun completed a study that confirmed the role for dominance. In their study, they found that pairs of Drosophila with large X chromosomes were able to develop HR faster than normal, which confirmed the role for dominance. The second theory that I'm talking about is the faster male evolution theory. And this explains that male genes are expressed to two different types of sexual selection, which are the male-male competition and female choice, while the female genes are only exposed to female choice. And because of the pressure placed on the male genes, it results in a quicker evolution. Genetic studies have also revealed that in the species there is more often male sterility genes than female sterility genes. However, Aves and Leptoptera are the exception. These groups often suffer more from male sterility than male sterility, so therefore they do not support the faster male evolution theory. Another promising approach could be to look for Haldane's rule in species that can be species that them undergoes sex that could stop the differentiation of sex chromosomes in the heterogametic sex. In these certain cases, it could possibly allow for the study to determine the primary causes of Haldane's rule. With the mapping of evolution of sterility and in males and females using phylogenetic trees, it may be possible to determine a detailed dissection of Haldane's rule.
So within chapter 5, there were two really big main points. And the very first one was dominance. So dominance does not alter the way that genes are inherited, but it is a result of interactions between genes of the same locus, which are allelic genes. So the first one is complete dominance, which means that the heterozygote has the same phenotype as one homozygote. So an example of this in humans is the brown eye color. So the brown eye color is dominant, which means that it only takes one brown allele to have brown eyes. So an example is blue eye color is also recessive. So that means if you have one brown allele and one blue allele, you're still going to have brown eyes, making it a complete dominance. The next kind of dominance is codominance, which means that the heterozygote exhibits traits from both of the parental homozygotes. So an example in humans for codominance is the MN blood type. So there are two alleles and one codes for the M antigen and one codes for the N antigen. So the homozygotes that have two alleles for the M antigen have the M blood type. And if you have two alleles for the N antigen, they have the N blood type. However, you can also be heterozygous and have one M and one N, and that would result in the MN blood type being codominant. And then we also have incomplete dominant, which is when the heterozygote has a phenotype intermediate between the two phenotypes of the parental homozygotes. So an example of this is if you look at flowers. So if you have a red flower crossed with a white flower, instead of only getting a red flower or only getting a white flower as your offspring, they can produce a pink flower, which is an example of the incomplete dominance. So the next main point that we looked at in chapter 5 was the sex-linked characteristics. So a sex-linked characteristic is a phenotype that is determined by the genes located on the sex chromosomes. So an example of this, as we've discussed before, is color blindness in humans. Then we also have sex-influenced characteristics, which means that the phenotype is determined by the autosomal genes that are more readily expressed in one sex. So example of this is in some goats, whether or not they have a presence of a beard. So the presence of a beard is a dominant gene in males, but it's recessive in females. So that means that males only need one allele coding for beards to have a beard, whereas females need, so it makes it much more likely for males to have a beard rather than the females. And then the last one is the sex-limited characteristics. So this means that the phenotype is determined by the autosomal genes whose expression is limited to one sex. So an example of this is in domestic chickens. So cock feathering is an autosomal recessive trait. So it's limited to only males. So all the females and some males will all have hen feathering, but however, the males can have cock feathering. So the genotype for both the males and females are the same. So they'll both be little h for coding for this. But since it is an autosomal gene, the males will have the cock feathering and the females will still have the hen feathering. So looking at why there's a relationship to Haldane's rule is that so they looked at how the infertility of hybrids is more likely to affect the heterogametic genders. So this basically means that one sex is more affected than the other. So this relates directly to the sex-limited characteristics. So in humans, the heterogametic gender is the male as well as fruit flies. But if we look at ducks, for example, and many other birds and fish, it's actually the female gender where they have the ZW as compared to the XY in our species. So understanding the dominance and the sex-linked characteristics set forth the groundwork for the development and understanding of Haldane's rule that we know today.
And these are just our references. Thank you.